Before watching this video, click on the subscribe button.
Bacterial staining techniques used in the microbiology lab. Visualization of microorganisms in the living state is most difficult challenge, not only because they are minute but also because they are transparent and practically colorless when suspended in an aqueous medium. To study their properties and to divide microorganisms into specific groups for diagnostic purposes, biological staining procedures in conjunction with light microscopy have become major tools in microbiology. Basically, there are two types of staining methods used in a microbiology lab. First one is simple staining and second one is differential staining. In simple staining, there is a use of only single stain for the visualization of morphological shapes such as cocci, bacilli, and spirali, and arrangements in which the chains, clusters, pairs, and tetrads. While in differential staining, there is a use of two contrasting stains. Differential staining is further divided into two groups. First one is separation into group in which the gram stain and acid forest stain are included. The second one is visualization of structures in which the flagella stain, capsule stain, spore stain and nuclear stain are included. Gram staining. Gram staining is a common technique used to differentiate the two large groups of bacteria based on their cell wall constituents. The gram staining distinguishes between the gram-positive bacteria and gram-negative bacteria groups by coloring their cells red or violet. The acid-fast staining is used to identify the acid-fast organisms such as members of the genus Mycobacterium. The cell wall of acid-fast organisms is resistant to most of the compound, thus they require a special type of staining technique. The Ziehl-Neelsen method is most widely used in the lab. Flagella staining. The flagella stain allows the observation of bacterial flagella under the light microscope. Bacterial flagella are normally too thin, thus the flagella stain provided modern coat to the flagella so they appear as thick enough to stain. Capsule staining. Capsule staining is a type of differential stain that uses acidic and basic dye to stain background and bacterial cells. In that way, the presence of capsule is easily visualized. Spore staining is a differential staining technique used to distinguish between the vegetative cells and endospores. The primary stain is malachite green that is used to stain the endospores, and water is used as a decolorizer, while the secondary stain, safranin, is used to stain the vegetative cells because they will lose primary stain, and endospores are resistant to restaining, so they will get color of a primary dye. The most common example is Bacillus species and Clostridium species. Nuclear staining. The nuclear staining is used for the microscopic anatomy and pathological material. It reports the dark color to the nuclei of the cells, while lighter color is the contrasting color to the cytoplasm and extracellular structures. All microbiological staining procedures require preparation of smears prior to the execution of any of the specific staining techniques that I have described in this video.